Okay, Sue? Orange one, orange one.
Okay, welcome. This is Susan Chambers, and I'm speaking from the National Museums of Scotland. And this is a beautiful image of a very well-known polychaete called Nereus virens, and it's undergone the first stages of its reproductive cycle, and it's what is called an epitoke. And this was taken from a living specimen in from a local shore around Scotland. Okay. Okay, so the title of my talk is What, Where and Why Worms, and I'd just like to briefly describe basically three reasons why I have looked at polychaete worms for over 30 years. And the first, as always with many things in life, it's really largely luck and opportunity about who you meet and who inspires you. And the second reason is that they're absolutely beautiful, and I don't think there are many jobs where you can say that what you do and what you look at is beautiful, and they are, but they are beautiful only when they're alive. When they're dead, they're not so wonderful. And then the third point is that because they are so extremely diverse and poorly researched, there is a lot of scope for for exciting work, and I have never ever run out of things to look at over the last 30 years. Okay. I'm just going to whiz through a few pictures or a few images of some very colourful worms, just for people who are not very sure what a polychaete is, and just so that you can be equally inspired as myself. This is Aphrodite. It's the sea mouse, and it's very common all around the shores of the UK, and it's got these iridescent bristles which Linnaeus described as beautiful, and which is why he named it after the goddess of love, Aphrodite. This is another very common scale worm that's found around the shores of the British Isles, and this was under a rock in Cramond. This is Circulid. The red tentacles are from the front end of the animal, and it makes these calcareous tubes which form reefs on some parts of the west coast of Scotland and Ireland, and they're absolutely stunning. This is another tube worm, a sabellid polychaete, that has again these beautiful tentacles, and it's common on the west coast of Scotland. And this is an image of Nereus virens, a whole animal of the head region of the first image I showed you. And this is taken from basically a polychaete farm that used to be active on the northeast coast of England, where these animals were bred for bait. This is Sabellariidae. These are removed from their tubes, and these also make reefs. This is a grapple worm, or Malgarnid, and it was found on the west coast of Scotland in the Inner Hebrides, and as you can see, it has these very large hooks. That's the wonderful thing about polychaetes. They have such a diverse range of characters, and particularly their chaetae. Another one, this is a whole image of Eulalia phyllodosid, again commonly found under rocks and stones around the coasts, and a spionid. This burrows in soft sediments. And this last one is a flavoliduride, and it was found in deep sea benthic mud from, I think, the Rockall Trough. Okay, so that's just a quick run through of the kinds of animals that we're talking about that are known as polychaetes. And so the next question was, where in the world? Well, because I work and live in Scotland, most of my work is in British waters, and that includes the continental shelf and deep water. And as we know, the seabed is 3 times the total land area of the UK, and more than half of Scotland is underwater. So this is the kind of area that most of my work is involved with. And as you can see, the large area called C7 which is just a name used for the Strategic Environment Assessment Area 7. This includes a lot of deep water, and where I have a lot of my samples are from there. So how do we collect them? I'm sure many people listening have been out on cruises or have been sampling, and this is just one of the ways of sampling. This is a grab from, I think, the west coast of Scotland again, and this is a trawl, and this is with Hamish from Heriot Watt, and this is a Van Veen grab, and this was taken from the Channel in the south of England, and this is a method that people are very commonly with, just shore sampling. But this is a special kind of a sledge that was designed by some researchers in Sweden, and it's called the Tjarno sledge because that's where they work, in Tjarno, the west coast of Sweden. And this sledge just scoops off the top surface layer, which is where most of the polychaetes live. Okay, once you have your sample of the benthos, it then involves sieving the sample. In this instance, it's not quantitative, but most of the time you're looking at quantitative samples. And then when you've sieved, usually on a half mil sieve, this is what you get. You get lots and lots of polychaetes, and in this occasion they were stained with rose bengal to make them easy to separate from the samples, from the sediment. Okay, one of the other questions I ask is, why do we need to know about marine worms? Well, as everybody is aware, there is an awful lot of disturbance of our marine environment through either hydrocarbon extraction or development, through fishing, through mining, through building harbours. There is a lot of disturbance, and most polychaetes live in the benthos. So in any benthic sample which I've just described, whether you take one from trawling or grabbing or just collecting on the shore, most of your sample, the largest part of that biomass, will probably be polychaetes, unless you happen to have a very, very large shell in there or a very large echinoderm, which will increase the biomass. But on the whole, the largest numbers will be polychaetes. Now, these numbers are not accurate because they're a very, very poorly understood group of animals, and there are approximately 1,000 species known from British waters and about 10,000 in the rest of the world, but that number is very debatable because of the poor amount of work that is undertaken. In this country there are only 2 people who are employed on taxonomy of polychaetes, and I'm one of them. And that means my job is supposed to be dominated by that. But we only really know the biology of distribution and detailed ecology of I would say about 20 species, maybe more, but only about 25, I would say. In order to illustrate the kind of work I do, I'm going to concentrate now on one particular taxon, and this taxon is called Chaetozone setosa, and Chaetozone is one genus within one family called Siraculidae, which is one of the 50 or so families of polychaetes in the world. So one genus, Chaetozone setosa, and I'm going to use this as a particular example. This animal is about between 1 and 2 centimeters long and 2 to 3 millimeters wide, so it's quite small. And to describe how I have arrived or even started the exploration of finding out more information about this animal, we need to go back to 1867. Chaetozone setosa was first described by Malmgren from Svalbard, sometimes known as Spitsbergen. And there was a good illustration, which is here on the right, and but there was no generic description. So basically he just drew it, said it was from Finland, Sweden and Spitsbergen, and that was it really. He didn't really say very much else about it. But then because it was quite a nice illustration, and his work was only one of very few works of polychaetes in the world at that time, it has been continuously recorded around the world for about 100 years. And as you can see from this black and white drawing, there's lots of chaetae, or bristles, in the front part of the body. And also you can see the tentacles there. Okay, so for the next 100 years, Chaetozone was recorded from the intertidal to 5,000 meters. It was described from all kinds of substrates, all grades of mud and sand, and then rock, coral, and even seaweeds. It was described from every ocean in the world. And this seems highly unlikely, although it's frequently referred to as a cosmopolitan species. So until the 1990s, there was only one species of Chaetozone in the world, well, not in the world, but known from the British Isles. I think another couple of species have been described from in the Pacific. And at that time, I began working with Annette Woodham, who worked for an environmental consultancy. And Annette realized that although she thought everything that they had been looking at for the previous 10 years was Chaetozone setosa, and they'd been calling it that because they had thousands of samples to work through, she began to doubt that that was all. She thought that there was something else in the samples that they had actually collected from the southeast of England, where a harbour was going to be built, and they needed an environmental impact assessment. So she collected several samples, and we began to look at them, and it was pretty clear that there was more than one species of Chaetozone present. And to cut a long story short, we called it Chaetozone gibba. And then we continued working, because as a result of that, we began to doubt what Chaetozone setosa was. And eventually we found another taxon called Chaetozone christii, and I'll come to that. And because of this, remember this is all quite slow because we were doing other things as well, but we were asked to look at samples from the Faroe Shetland Channel. And this, as you know, is deeper water. And it was a whole different area for us. And when we started to look at these samples, we found that there were many, many more taxa than we'd ever seen before. And we called one of them Jubata because we only had samples in good condition from a few sites that were whole and that we could describe. And that's why we only described one. So just to show you what these were, the image on the left is, as I've said, is setosa. The next one to it on the right is the one that I called christii, and then the next one is gibba, and the last one is jubata. And I hope that you can see, just by looking at these whole animals and looking at their general morphology, that they are quite different. You don't need to be brilliant to see that their body shape is different. And if you look at Chaetozone setosa, on the left you can see lots of chaetae. The next one, christii, doesn't have any long chaetae. And the next one, gibba, doesn't really have long chaetae. And also, I know you can't see it from here, but gibba has eyes. And I'll come back to that in a minute. And then the last one is jubata. And although it has long chaetae, they are all at the front. And jubata means lion's mane. And as you can see, it's just sort of, although the chaetae are at the anterior end, they're of a quite different distribution to setosa. Now, we don't only look at morphological characters. In order to do this separation, what we had to look at was many publications and collect lots of data. And this involved looking at thousands and thousands of specimens that people had called Chaetozone. Now, when we looked at christii, when we gathered all the data together, we found that it didn't really occur any deeper than 20 meters, occasionally 50, but that was a bit odd. But on the whole, all the samples were from less than 20 meters. It was intertidal to about 20 meters. Gibber was pretty much the same. It was intertidal about 20 meters. But gibber was found in more shelly kind of gravel, whereas Christii was found in coarse sand. And of course, Jubata was found in very deep water. I think the shallowest we ever found Jubata was 1,000 meters. It was between 1,000 and 2,000 meters. Now the other thing that led us down this path of looking at these different characters was really the reproductive information. And there was a fantastic publication by a PhD student from Newcastle, from the Dove Marine Lab, and he was in fact called Graham Christie, which is why one of them are called Christii. And Graham, he wasn't really looking at the taxonomy or the morphology, he was really looking at the reproductive biology. And in his PhD and subsequent publications, he called everything Chaetozone setosa, which is perfectly understandable because that wasn't the focus of his work. His focus was the reproductive biology. And what he'd found was, he said he had 3 groups of animals, and they all had different egg sizes, and they bred at different times of year, and they lived in mud. Some lived in mud and some lived in sand. And I thought, this is very odd here. Something is odd. You don't get animals that are living in mud and breeding at different times of year in sand and all those complications. And when I came to look at them more clearly, what I came to see, and when I borrowed samples of his from the same sites that he'd been to, some offshore sites at 50 meters and below, which was called Station P, and the other sites which were shallow, they kind of fell into very nice clear patterns. That those that lived in the coarse sand and bred at one particular time of year, I think it was March, were all Christii. And those samples that lived offshore in mud and bred in November were all setosa. So although he did have setosa from his offshore sites in mud at Station P, the other samples that he had were Christii, which is the second one next to it on the right. But he didn't have gibber, but he had another taxon, which I haven't described yet because I haven't got enough samples that are whole. But in that taxon, the shape of the sperm are completely different to anything else, and I haven't got around to doing that yet. But the gibber, which he didn't look at, he didn't see because it doesn't occur in northeast England. So you can see it isn't just the morphological characters that you need to look at, but they help you find a way in to start, and then you can look at other things. So what Annette and I did over the next 10 years was we started plotting numbers of segments, again, length of the animals, again, and it's morphological characters. But with an MSc student at Heriot Watt, we also started to use GIS, and we started to plot this information, and it took an awful long time. But as you can see from this map, the crosses, which we'll talk about first, there's the one in the Faroe Shetland Channel and the other one which is just off Norway. That is Jubata. They're the only sites that I have Jubata from. And although the site off Norway is not particularly deep, it is very cold water. And that seems to be a factor in the distribution of these particular species. So when we plotted the sediment type with the temperature, we have found a very nice pattern. And as you can see, all the circles with like the bullseye with the dot inside are setosa, and that doesn't really occur any further south than the southern North Sea and the Bristol Channel on the west side. And the triangles are Christii, and although it appears to overlap with setosa, it only occurs in shallow water, and as you can see, all the triangles are really quite close to the coast or some island, and even the record that's off Iceland there, that's quite shallow water there. And the inset with Svalbard are all Chaetozone, which is no surprise because that's where Malmgren described it from, and that's quite cold water. Now what we found when we plotted these were that it seems that Chaetozone doesn't like water that's warmer than 10 degrees in the summer, and Christii doesn't like cold water. It really only seems to reproduce in warmer water where there is temperatures of about 12 in the summer. So again, it's adding another layer of information about their distribution, and may help towards just trying to work out how diverse this genus is. Now, when I published this publication about the distribution of Chaetozone, I had quite a lot of correspondence, and I had a lot of requests from people telling me that I was wrong, and that they did have Chaetozone, and it's all around the Mediterranean and off West Africa and everywhere. So I asked people to send me their specimens, and usually I find that they're not Chaetozone. They're always either Christii or Gibber or another genus called Coriellus atlantica, or an undescribed species. And although I've been very reluctant to move out of my comfort zone in the North Sea and the Northeast Atlantic, I have recently become involved in looking at samples from the Mediterranean, because there are so many people working out in the Mediterranean, and there are so many problems that I kind of got sucked into redescribing another species of Chaetozone from the Mediterranean. But meanwhile, I'm trying to concentrate working in the North Sea again and the Northeast Atlantic. And since I've worked on those publications with Chaetozone and Gibber and Chaetozone, I have found these other taxa. And the one on the left is Chaetozone D at the moment, and it's very common. And it became confused with Chaetozone because it's quite long, and people thought it had long bristles and all kinds of other things. It occurs in mud and it's shallow. So that has been confused with it, but that needs redescribing. And the other two drawings are of very deep water animals, which were along with Jubata, and hopefully one day I will get around to describing those as well. But at the moment I am working on another genus which is very similar to Chaetozone called Monticelina, which has been confused with this Chaetozone D. And so it's kind of unraveling another part of the jigsaw. And so what has happened over the last 20 years that I've been working on Chaetozone is that originally we thought we only had Chaetozone Chaetozone from the British Isles, but now we have at least 6. And as I mentioned before, Chaetozone is only one genus within the Siraculidae, which comes back to how we can't be so sure that we only have 1,000 species of polychaetes in the UK. It's probably many more. It just depends how much you look at them and how many people there are. And within the Siraculidae there are 8 other genera, and I know they all require the same amount of attention. But it's taken me at least 10 years to get this far, so I'm going to run out of time before I get around to the other 8. It isn't just the morphological characters that I look at. I'm also very interested, and I'm working with other people to look at the molecular data to see if this helps at all. Usually I find it confuses the ideas, but that's a good thing. But we also need to look at the reproductive biology of all these other taxa. At the moment, the only work that's been done was done by Christie in the 80s, and that was on Chaetozone. We also need a lot more very good ecological data. Most of the data I get is quite poor. I need very specific data about the sediment and the date and the temperature, and hardly anybody ever records temperature. So there's quite a lot of work to do in just gathering this data and pulling it together so that we can be sure about exactly how diverse this group of animals is. And the reason that many people need to know is for policy and for deciding on which areas are worthy of conservation, which areas are not. They need accurate figures. And at the moment, it's very, very slow to provide this information because it's not easy. It actually takes an awful lot of time. The other absolutely wonderful thing about my job, apart from looking at beautiful animals, is I get to go to beautiful places. And this is Tjarno off the west coast of Sweden from the accommodation where I was staying. And this is the ship that we went out on to collect our samples. So it's a wonderful job, and I'm extremely lucky, and I'm happy to take any questions. Thank you very much.
Okay, thank you, Sue. We'll just now take a moment just to make sure we've got everybody else's attendees on our list and to see whether anybody's put their hand up. Can we go to attendees? Yeah. Okay, do you want to open the microphones up? Just unmute.
Just do it individually.
We're just unmuting everybody at the moment. Just literally unmute. There, that's it. Okay. Everybody should now be unmuted, and we're open to any questions that come in. Obviously, this is the first time we've done this, so hopefully people won't talk over each other. So if anybody has a question, if they'd like to raise a hand or just ask us directly, and just give us your name before you ask the question. Okay, I've got one, a bit like chairperson's privilege, in the sense that, Sue, I know that a lot of work was done by PJ Olive in Newcastle looking at various aspects of polychaete biology, and more recently looking at its uses in aquaculture and also other biotech uses as well. Can you just tell me what your understanding is of the status of that work? I've not really been keeping pace with it, but I know there were certain interesting uses and compounds that were being made, or could potentially come from polychaetes. Can you hear me, Sue? Sue, can you hear me?
Yes, I can hear you now.
Did you hear the question?
Yeah, who was the person who you said did the work?
PJ Olive at Newcastle.
Oh yes, Peter Olive, yeah. So what was the question? What was the specific part of that?
He seems to be doing quite a lot of work using polychaetes for various different purposes, including aquaculture, diets for prawns and shrimps and things. And I know that they set up a company to do that, but they're also beginning to look at some of the other potential biotech uses, in that I think somewhere along the line discovered that they were producing, or potentially de novo producing omega-3s in their guts. But all that work went very quiet, and I don't know whether you had any updates on that.
Yes, I think that as far as I know, they still do an awful lot of work on reproductive biology there, but their aquaculture enterprise was run as a separate business, and then it was sold on to a company called Shoreline, and I think they went into liquidation last year.
So in terms of your taxonomy then, are there any, I suppose in a way, key species that we should be looking at as biological indicators that you need to focus on in terms of the taxonomy?
Yeah, well, one of them is Chaetozone setosa, because it becomes very common in disturbed sediments. So there's Chaetozone, which is why I got involved with it, because so many people were doing environmental impact assessments and finding Chaetozone everywhere, which they called Chaetozone setosa, although we now know it isn't. It isn't always that, but it is one of the many. And the other groups that are also very, very common for it when they're examining samples from environmental impact assessments are spionids and dorvidelids. There's quite a few taxa, but not many people working on them.
Okay. And in terms of, if you like, molecular taxonomy, is that really the way forward, or is there still really a place for, if you like, some of the historic morphological work, or is everything becoming omics now?
I think it's a very interesting tool, and I think it's an addition to all the others that we use. We still need the ecological data, the reproductive data. It's just another part of the bigger picture that we're adding to the information that we have about our diverse fauna.
Are there any other questions out there from the audience? Obviously it's a bit difficult to see hands raised at this end, but certainly if anybody would like to ask a question.
Yes, this is David Ferry. I was wondering, with regard to the molecular data that you were talking about then, you said in your talk that it confused things, or it conflicted a bit with some of the morphological or ecological data.
Yeah.
I'm wondering if you could expand on that a bit, please, and in what way does it confuse things?
I think, as you know, what you need at the beginning is you need your samples to be of what you think is one taxa. Whereas if people are collecting them and they're confusing and they're putting more than one taxa within their molecular data, then you get a confused answer. You need to be very clear that what you have at the beginning is one thing. So I think I mean, I don't know.
Initial identification is the problem rather than the molecular data per se.
Yeah.
Okay.
Yeah. Yeah. Because it just I can't give a particular well-known example with polychaetes because there isn't that much done really. I mean, there is some, but not much. But I know, for example, with leeches, people have looked at the molecular data for leech and they wondered why it was all confused and it was all, it didn't mean very much, and there was a lot of work done on this because obviously medicinal leeches became quite important. And then somebody went around the labs and looked at where they were breeding them and found that they'd got at least 5 different taxa in the labs around the world, and that's why it was confusing.
Good, thanks.
Somebody obviously has birdsong in the background, which is rather nice. But, are there any other questions? Okay. Well, really, I think that draws it to a close, really. Really just to remind everybody that on the 1st of July, Martin Cox from the Scottish Government will be giving us another webinar, and this will be about the National Marine Plan Interactive, which some of you may be aware of. You can dabble with it on the online system that they have. If you type in to Google NMPI, that's capital N, M, P, and a small I, it will take you into an interactive online database, which is GIS based, and there are lots of interesting layers on that, and Martin will be discussing how that's going to be developed in the future and what it's being used for at the moment. And that will be on the 1st of July, again a lunchtime webinar, and we'll be advertising others in the near future. I think this has been reasonably successful, and very many thanks to Sue for being the first guinea pig on this. So thanks again, Sue. I think we'll all sign off now and go and enjoy some lunch, and we'll see you in the next webinar. Thanks very much. Bye-bye.
Bye.